Hello students, welcome to EPG Pathshala. I am Dr. Imtiaz Hassan from Jamia Millia Islamia, a central university. Today we are going to talk on module binding studies. Here we discuss strong binding, strong two binding sites, weak or single binding, weak two binding sites, cooperativity in binding, competition dialysis assay under the paper techniques used in molecular biophysics 2. After this lecture, the student should understand binding studies, how the binding studies performed, what are the parameters used in the binding studies, types of binding studies such as a strong single, a strong two binding, weak single, weak two binding site. Then properties that affect the binding of the protein and ligand. And then we talk about the cooperativity in the binding, and at last we discuss the significance of the competition dialysis assay. So let's discuss protein ligand interaction. Interaction of ligand with receptor is known as binding, which can be specific or non-specific. A ligand is molecule that recognized by the protein or receptor and is able to bind to that receptor. The site at which ligand bind to a protein is called ligand binding site. And the ligand binding site of protein is quite flexible, making it easier for the ligand to bind by inducing some conformational changes. The ligand binding sites are of course complementary to the protein to which it binds or to the ligands. Reversible binding of ligand is essential, and specificity of ligand is similarly important because the ligand binding is often coupled to the conformation changes. Sometimes it is quite dramatic as by induced fit. In multi subunit protein, conformation changes is in one subunit can affect the binding of to the other subunit, which is called the cooperativity, and it is highly regulated process, such as the example of the hemoglobin, antibodies and muscle contractions. Here we try to show you that the binding studies between protein and ligand. You can see here the protein, simply receptor, is of bigger size, which where the ligand can bind, and this model is showing how the ligand bind to the protein molecule. So it is a reversible transient process of the chemical equilibrium between A and B that leads to the formation of AB complex. A and B is refers to the protein and ligand respectively. The molecules that bind to protein is called ligand, typically a small molecule. The region in which the protein where the ligand bind is called the binding site, and ligand bind via some non-covalent forces that dictates the protein structure and allows the interactions to be transient. A molecule changes from higher potential energy to the lower potential energy form, and potential energy can be determined by intermolecular and intramolecular and environmental forces. Now it talks about the ligand receptor binding. Ligand binding is the interaction that occurs between two molecules such as protein binding as well as the degree of the affinity, either the weak, strong, or there is no connection between the binding affinity with the binding site for which the reactant bind together. The essential aspect of the binding includes but not limited to the concentration level of the reactant or product and maintaining the equilibrium constant of the reactant throughout the assay and the reliability and the availability of the linked reaction. Assay of the binding are very simple, although it fails to provide the information on whether or not the compound being tested or affect the target function. Here we discuss about the ligand receptor binding. You can see here that the ligand bind to the receptor from the ligand receptor complex, where we measure the parameter like the binding affinities, which is dependent on the intermolecular forces and leading to the conformation changes. So the technique is used to estimate the ligand receptor binding is based on the biological response observed by the binding of the ligand to the receptor. So the receptor are sometimes are globular proteins located mostly in the cell membrane and receives message from the chemical messenger coming from the other cells. It transmits a message into the cell leading to the cellular effect, and different receptors are specific for different chemical messenger. Each cells have a wide range of receptors in a cell membrane, making it responsible for different chemical messenger. The binding affinity of the ligand and receptor is depend on the types and the number of intermolecular forces. These forces are covalent bond, that is very strong, and its bond energy is 5 to 50 kilocalorie per mole. Normally irreversible binding. It involves in normally irreversible binding. The other forces are non-covalent forces are ionic bond, which having 5 to 10 kilocalorie per mole. Hydrogen bond, it has the bonding energy between 2 to 5 kilocalorie per mole, and hydrophobic interaction, which having the lowest binding energy. And all three are of reversible in nature. Here we discuss about how does the binding site changes the shape. You can see here the site of receptor where the ligand bind and it induces some conformational changes. So before the intramolecular bonds not optimum length for maximum binding strength, however after it induces the intramolecular bond because of its lengths are optimized. So the binding interaction must be strong enough to hold the messenger sufficiently long for signal transduction to take place. Interaction must be weak enough to allow the messenger to depart and implies a fine balance. Designing molecules with a stronger binding interaction results in drug that blocks the binding site antagonist. Here we discuss about the agonist and antagonist. Agonist activates receptor to produce the desired response. Conventional agonist increases the proportion of activated receptor. However, the inverse agonist stabilizes the receptor in its inactive conformation and acts similarly to the competitive antagonist. Many hormones, neurotransmitters such as acetylcholine, histamine, norepinephrine, and drugs. Example: morphine, phenylmorphine, isopritazole, and benzodiazepine, barbiturate, etc. So you can see here how the agonist bind to the protein receptor and bring about the appropriate response. Agonist influence increases the response, produce the positive response. Antagonist reverse the value of agonist, and some are the inverse agonist that gives the negative response. Now we talk about the agonist and antagonist. Antagonist prevent receptor activation. Antagonist increase cellular function if they block the action of a substance that commonly decrease the cellular function. Antagonist decrease cellular function if they block the action of a substance that normally increase the cellular function. So receptor antagonist can be classified as reversible or irreversible. The reversible antagonists readily dissociate from their receptor, and the irreversible antagonist forms a stable, permanent or nearly permanent chemical bonds with their receptor, mainly by alkylation sometimes. The pseudo-irreversible antagonist slowly dissociate from their receptors. In competitive antagonism, binding of antagonist to the receptor prevents binding of agonist to the receptor. So on the other hand, in non-competitive antagonism, agonist and antagonist can be bound simultaneously, but antagonist binding reduces or prevent the action of agonist. In the irreversible competitive antagonism, agonist and antagonist form short lasting bonds with the receptor and steady state among agonist, antagonist and receptor is reached. Such antagonism can be overcome by increasing the concentration of the agonist. You can see here in the graph. Here we talks about the types of binding sites. There are two types of the binding site. One is the single binding site where the one ligands bind to one receptor and the stoichiometry is 1:1, and the binding equation is given as K1 for the forward reaction, K2 for the backward reaction, and it follows the simple Michaelis-Menten equation. However, the multiple binding sites, there are maybe multiple ligand, one receptor and one multiple receptor, one ligand, and it follows the different kind of the equation, that's the substrate inhibition, where the two binding sites can be taken in account. You can see here for the binding affinity, the equation, for example, the protein P and the ligand A, it forms the PA complex. So the association constant for Ka is given as the concentration of Pa complex divided by concentration of P and concentration of A, and the dissociation curve constant Kd is the reverse of the same equation, that is the 1 by Ka. So rearranging this equation, you can see Pa equals to Ka into P into A. So V represents the moles of bound ligands per molecule of the protein. So V equals to Pa by P equals to Pa by P plus Pa. Concentration of P and A represents unbound protein and ligand respectively. The protein total and ligand total represents the total protein and ligand in the system. So V equals to Pa, concentration of Pa divided by concentration of P plus Pa equals to Ka into P into A divided by P plus Ka into P into A, that's equals to Kd into A divided by 1 plus Kd A. By rearranging this equation, we can get the value of V, which is the equals to concentration of A divided by Kd plus concentration of A. In many experimental procedures, we can measure velocity V and A at a range of ligand concentration, which allows you to estimate the value of Kd, and from the delta G, and from it you can calculate the delta G of the binding as well. It represents the binding affinity, where the binding affinity V is equals to A divided by K D into A. A is the concentration of unbound ligands, which is the same as the total ligand concentration. Certain binding experiments involve the measuring the concentration of bound ligand Pa instead. So in this case, you can simply easily calculate value of A equals to A total minus Pa. It may also be that A equals to A total. So it is true when concentration of A is very high, very greater than P. So biological system can be easily described as having constant pressure P and constant temperature T. The system is free to exchange heat with the surrounding to remain at a constant temperature. It can be expand or contract in volume to remain atmospheric pressure. So the V equals to A divided by Kd plus A, or the Kd equals to P into A divided by Pa equals to 1 by Ka. So the Kd is the concentration and has units per mole per liter. Kd gives the concentration of ligand that saturates 50% of the site when total site concentration is much lower than Kd. Almost all binding sites are saturated if the ligand concentration is 10 into Kd. The dissociation constant Kd is related to the Gibbs energy delta G by the relation delta G equals to minus RT ln Kd. Here we talks about the binding affinity. If instead we have multiple identical binding sites, then P plus Na equals to PAN. Then it is possible to show the appropriate form of the Scatchard equation V by concentration of A equals to N into K alpha minus K alpha into V. So the plot of V by A versus V should be gives the straight line of binding reaction with identical site. The slope of the plot is minus K and Y intercept is NKA. So the increasing complexity of the binding sites are equivalent and independent, may be cooperativity or the heterogeneity at binding sites are the equivalent and non-important. In the heterogeneous, the binding sites are independent and non-equivalent. Heterogeneity and cooperativity at the binding sites are not equivalent in the independent. So the simple to very difficult process can take place, and this Scatchard plot based method is used to estimate the binding affinity in the complex system. Here we talks how to understand the binding affinity. You can see here in the figure that the strong binding and weak binding, strong binding large number of interaction takes place, however in the weak binding lesser number of interaction takes place, and the value of Kd can be used to determine the affinity of a drug to its receptor. Affinity is the strength of interaction between the ligand and its receptor. High Kd means weak interaction, low affinity, however the low Kd means a strong interaction, high affinity. As the concentration of free drugs increases, the ratio of concentration of bound receptor to total receptor approaches unity. Here we discuss the properties that affect the binding. The binding of a protein ligand is largely affected by the complementarity, flexibility, surface area contact, non-covalent forces, and of course affinity. So these are the major factors that affects the binding affinity of the protein ligand interactions. Now it talks about the cooperativity in the binding. Cooperative binding is observed in many biopolymer including protein and nucleic acids. Cooperative binding has been shown to be the mechanism underlying the large range of biochemical and physiological processes. The simplest phenomenological model that account for cooperativity is ligand binding to the macromolecule with two binding sites. The cooperativity in binding represents perhaps one of the most interesting and not fully understood type of molecular interaction observed in nature. For macromolecules having two or more binding sites, cooperativity is characterized by a change in intrinsic or site-specific equilibrium binding constant as a function of other reaction progress, that is the affinity of the given binding site for a ligand will be affected by the occupancy of the other sites by the same or different ligands. The first modeling approaches describing the cooperative binding were proposed by A.V. Hill at the beginning of the 20th century by analyzing the binding of oxygen to the human hemoglobin. Here I try to show the cooperative binding. Here you can see the binding site, two binding site, and binding to the one site is affecting the binding to the other site. So that's what one binding is leads to the cooperates with the binding to the other sites. It is known as the cooperative binding. Cooperativity in binding, here we consider the impact of cooperativity on metabolic reaction in the case of inhibitor binding to the enzyme. In this situation, binding at one subunit can influence the affinity of the other subunit for the same inhibitor, thereby affecting how the rate of catalysis responds to the inhibitor concentration. The red curve shown positive cooperativity where inhibitor binding to one subunit causes additional subunits to have higher affinity for the inhibitor. And the blue curve shown the negative cooperativity where the inhibitor binding causes other subunits to have a lower affinity. Here you can see the myoglobin and hemoglobin polypeptide. Although the both are having the similar kind of chain, polypeptide chain, only the difference is that the hemoglobin is having many subunits, 4 subunits. However, the myoglobin having the single subunits. That's what the hemoglobin binding of oxygen to hemoglobin is a cooperative process rather than the myoglobin is the non-cooperative process. Here we show the changes in alpha 1 beta 2 and alpha 2 beta 1 interface into T2R state transition in hemoglobin. You can see here the structure of how the structure and conformation of a particular amino acid changes from T2R state or the deoxy to the oxy state of the hemoglobin. The changes in the hemoglobin quaternary structure with the oxygen binding. You can see here that some major changes occur when the oxygen bind to the hemoglobin. Rotation of alpha 1 beta 1 relative to alpha 2 beta 2 and change in the size of central cavity, shift of C termini and FG corner of beta chains relative to C helices of alpha chains. C terminal of beta chains interact to the C helices of the alpha chains in T-deoxy state. These interactions are broken in the transition to R-oxy states. Is showing the structural transition between T to R state. You can see here the structure of the T state is totally different from the structure and conformation of T state is totally different from the R state because of the cooperative binding of the oxygen. Oxygen binding curve. You can see here in the T state and in the R state different types of interaction takes place, and if you plot the value of theta versus partial pressure of oxygen in the low affinity state and in the transition from low to high affinity state, and then you can see the high affinity state. That's because of the cooperative affinity of the cooperative binding of the oxygen to the hemoglobin. Here we discuss about the hill plots. The slope of the region of the 50% of saturation is called hill constant. Here the log theta divided by 1 minus theta is plotted versus the log of the partial pressure of the oxygen, and you can see here the different if depending on the number of binding sites, the different myoglobin it having the NH number of hills is 1, and the hemoglobin high affinity state 1, and the hemoglobin NH equals to 3. So the slope of the 50% of saturation is called Hill constant, where N is more than 1 positive cooperativity, N equals to 1 no cooperativity, while the N is less than 1 means negative cooperativity. For myoglobin NH equals to 1, it indicates non-cooperative process. However, for the hemoglobin NH equals to 3 to 3.5, this indicates positive cooperativity since NH is more than 1 with at least 4 binding sites for the oxygen. Intercept with the broken black line at 0 value for the log theta minus theta 1 indicates the partial pressure of the P50, and so the oxygen binding affinity is lower P50, higher affinity. Hemoglobin high affinity oxygen binding log P50 equals to upper asymptote of the intercept. However, the hemoglobin low affinity oxygen binding log P50 lower symptomatic intercept. Slide is representing simply the two mode of allostery. Here you can see that how the binding of oxygen to one subunit cooperates with the binding of to the other subunits and ultimately all subunits in a cooperative manner or in an allosteric manner. Again, we are showing the two model of the allostery. The one model is the Koshland-Nemeth-Filmer model given in 1996 by the indicating sequential or induced fit model, while the other model is by the Monod-Weidman model, MWC model, that concerted or the asymmetry model. Competition dialysis assay is a powerful tool for the discovery of ligand that bind to nucleic acid with structure and sequence selectivity. This method is based on firm thermodynamic principle and is simple to implement. In the competition dialysis experiment, an array of nucleic acid structure and sequence is dialysed against a common test ligand in the solution. And after the equilibration, the amount of ligand bound to each structure or the sequence is determined spectrophotometrically. Since all the structure and the sequence are in equilibrium with the same free ligand concentration, the amount of bound ligand is directly proportional to the ligand binding affinity. In competition dialysis assay provides a direct and quantitative measure of selectivity and unambiguously identifies which structure or sequences within the same sample array that determined by a particular ligand. Here we describe the competition dialysis assay. Here we simply put the DNA molecule in the dialysis bag and keep it in the different types of the ligand. So AT is the total amount of substrate, AB is the amount of bind substrate, and A is the amount of unbound substrate. So A total equals to A bound plus A unbound. It is used to detect physical changes in the binding, delta H, that is change in the measured quantity, and delta St is the total change in the measured quantity. So the fraction of site is given in a proper equation, and that's the whole equation is derived from protein to DNA, protein P to DNA fragment, and by using the whole equation we can be able to estimate the binding constant or dissociation constant of the any DNA ligand interactions. Here we try to show the stoichiometric titration to determine the number of binding sites. To a solution of DNA interacts with a single binding site, a small amount of protein P are added. Since the binding site affinity for protein is high, low Kd values as compared to the total DNA concentration, practically every protein binds as long as there are free binding sites on the DNA. This is termed as stoichiometric binding on stoichiometric titration. So students, let's now summarize what we have learned in this module. Properties of protein that affect the ability of enzyme to bind to it are its flexibility, complementarity, surfaces, and non-covalent forces. The flexibility allows an easier fit between the binding sites and the enzyme, and the complementarity and the surfaces are important factors that contribute to the specificity of enzyme to the binding site. It may be assumed that covalent forces are used due to their ability to better bind to the enzyme to its active site. However, the strong binding forces of covalent bond makes it too difficult for the active site to release the enzyme. It must be kept in mind that the enzyme do not bind forever in the active site, and as a result, non-covalent forces are the best for easy recognition for the substrate and releasing it. Cooperative binding occurs in binding system contains more than one type and or a species of the molecule in which one of the partner is not monovalent, and it can bind more than one molecule of the other species. In final, the competition dialysis assay is a simple and straightforward tool for the discovery of ligand that bind to nucleic acid and with a structure and sequence selectivity. Thank you.